Hey team, welcome back. Last time we talked about peptidoglycan structure, how it's made, all these lovely steps. We talked about lots of enzymes involved and how bacteria can respond to antibiotics that interfere with this process. But today we're going to still be talking about the cell wall, but we're going to talk about how the structure of the cell wall can define the shape of bacteria. And there's a lot of detail involved with the division, elongation, and curvature of different bacteria. So let's get started. Okay, so once again, I'm just gonna write cell walls. This is still what we're talking about. If you want, you can say you can add the detail of, we're gonna be talking about division, elongation, curvature, but we'll be writing that as we take the notes. So I feel like it's not entirely necessary. Okay, first, before we get into the deep dive, let's go into a basic review in case you aren't familiar with the different structure of bacteria. So there are kind of three shapes that we mostly learn about. Bacterial shapes. We have cocci, and these are spherical. Pretty easy. The only step involved in making cocci bacteria is division. So that's pretty easy. You get bigger and you divide, and then there's two of you. Awesome. What about rod-shaped bacteria? Obviously, rod-shaped. So these have division too, you know. That's how you make more. But then they have elongation. So they'll divide like this, and then there'll be two small pieces, and then they'll elongate, and then they can divide again. And then our final one are spirally ones, so spirale / vibrios, like Vibrio cholerae, cholera. And these are going to be kind of topsy-turvy. And so this has also division and elongation. But it also has curvature. Curvature. Okay, it fit. Okay, so let's talk about each of these steps. Division. So first things first, we need to make more peptidoglycan. If we're dividing, there's going to be two of us now. We need to make this. You see how there's nothing there? Now there is. So we need to make that. Make more peptidoglycan. And so we'll have our PBPs that we talked about last time and SEDs that do that. Awesome. So when we go from one bacterium to another, we're going to want a septum to form in the middle here. So we can draw kind of like that. It's not, it's kind of more like this. Okay, so as we're dividing, we need this to form. Septum. And that's what we need our peptidoglycan for. So how did we find the genes that did this? So as we kind of covered in this class before, we're going to want to look for a conditional loss of function. So here we're looking for temperature sensitive mutants. And we can abbreviate that with FTS. Because we're fission, we're breaking into two. These temperature sensitive mutants will behave differently at different temperatures. So if we have 30 degrees Celsius and 42 degrees Celsius, if we function normally at this temperature, maybe we'll have bacteria that look like this. And this is what they found, actually. I'm not just saying maybe, this is what they found in these specific fission temperature sensitive mutants. At 30 degrees Celsius, they function normally, but at 42 degrees Celsius, they were long. So they're still making peptidoglycan. In the lateral cell wall here. But we're not making the septa that we need to divide. So we have a problem. So what genes do we think were changed in these mutants? Well, we actually already know of a couple that are involved in division, and we talked about them in the last lecture, and obviously they didn't know about these when these experiments were being done, but we don't know about them, so we can probably guess what might be involved. So we know that PBP3 acts at the division site, right? Well, they found the gene that encoded it And this is a transpeptidase, right? All of the PPPs are. And the gene that encoded it was FTS1. Encodes PBP3. Oh, PBP3. And there was another protein we talked about last time, FTSW, and that's one of the SEDS proteins, and we'll talk about that in a sec, but first let's talk about FTSZ actually. FTSZ. So this is the first thing to arrive at the division site. And it recruits other proteins to form the divisome. Oh my gosh, divisome. Which is the complex that makes these septa by synthesizing the cell wall between dividing cells. In dividing cells, it creates this so-called Z ring, because I guess it's FTSZ. And this just kind of marks the division site. So imagine this is like a 3D structure, like this side of the ring is towards us and this is back. So we can say Z ring. Cool. So let's talk about what's part of the divisome. So if there's other proteins involved, let's talk about the one I was just mentioning that SED we were talking about. So now we get to talk about FTSW. That's the SEDS protein we were talking about before. That's our transglycosylase, as we learned last time. We also we need a transpeptidase too, right? So we recruit this PBP3 FTS1. So we can say either, I guess. Okay, so PBP3 or FTS1, that's our transpeptidase. And we have some other components too. So there's ZIP-A and this is probably the gene, right? FTSA, which stably attaches this complex, the divisome, to the membrane. And then we're building peptidoglycan, right? So we need to have enzymes that make peptidoglycan. Murray. What about lipid 2? MERG. So PG synthesis, because how are you going to be making a cell wall without any peptidoglycan? And then one more piece, which is FTSK. And this helps finish evenly dividing DNA across that cell wall. This new septum. So clearly there's a lot going on here in the divisome, making that septum in division here. Lots of different things going on. But if you're studying this process, you might be wondering, if you're familiar with the way that animal cells divide, they have a cleavage furrow, right? Like they have it looks like a butt and it just divides. And plant cells have this cell plate where the cell wall kind of gets delivered into the middle until basically you have two cells. How does it happen in bacteria? Well, researchers were trying to figure out. And the way that they did this is they gave the cell a fluorescent dealanine, right? Okay, so this is, imagine a fluorescent dealanine derivative, actually. And so they could see the way that the cell wall was being made. Because it's fluorescent, they can see it. And at different times, they switched out the color. So I'm drawing it kind of how it's actually being built. They just saw the end product, but they know which colors they use first. So they use this color first over several stages, and they saw how the peptidoglycan was built. So the second color they used would be like this darker brown. And so since they were recording, which colors they were using in what times, they could see the order of how the peptidoglycan was being built. And so this would be like our third color. So with different colored dealanine derivatives, they could see, oh, the cell wall is being built inward. So between like two bacteria that are dividing, right? If you were to look like, okay, if the division site is there and you were to look, like if this is your eye, you have lots of eyelashes and you're looking like this way, right? Into this like ring that's happening here, it would look like that, right? Do we see the vision? Like this is kind of a ring, like before with this one. If we were to look into that, this would be the inside, and this was built last. So as this, because it makes sense that it would be like going from the middle outward because there's already wall here, so the wall kind of like goes in. Very interesting. So that's basically what we need to know about cell division in bacteria. Let's do some review. So our bacterial shapes, we have cocci, rod, and vibrios and spirale, which each add a step. There's division, elongation, and division, elongation, and curvature, which are like kind of, cocci. There's like, it's a very round word. You got some C's, they're round, and no, that's round. Rods are rod shaped, that's easy. And then spirale is like spiral, and then vibrio, I just know that cholera is squiggly, so I don't know, remember that. And then we got to make more peptidoglycan, so we use our PPPs and SEDs that we learned about before. And then we wanted to learn what genes do this, and so we used fission temperature sensitive mutants, our conditional loss of function mutants. And we saw two different temperatures, two different phenotypes, where this long one was not making septa, still making peptidoglycan. That's how we still got more cell wall, but they weren't dividing. And so we learned what things were involved in that. So we have PBP3, we knew that before, and we knew that from the previous lecture, but then they found out what gene encoded that. And FTSZ, very important for the formation of the divisome, that complex with all the stuff that helps you divide. Cell wall between dividing cells. We have our FTSW, which is a SEDS protein, which is our transglycosylase. FTS1/PBP3, so FTS1 meaning the gene and PBP3 meaning the protein. I don't know, we can underline both of those. Those are both important. That's our transpeptidase. Hopefully we're remembering that from last video. And then ZipA and FTSA, which both are attaching the complex to the membrane. And MRE and MIRG, we remember this from before, right guys? Right? And then our new guy, FTSK, which helps make sure we have an even amount of DNA on both sides of the cell. Like our both new cells have an equal amount of DNA, because it kind of suck if you didn't have all your DNA. And then by using different fluorescent dealanine derivatives, I'm just going to say different color. These are all labeled using different fluorescent dealanine derivatives. We could see that it's being built inwards. Septum is built inward. Okay, cool. Well, that's really interesting about division, but like I said, we have to talk about division, elongation, and curvature. So let's talk about elongation. And you can't draw on this side of the page because then you'll see all the bleed through and you're going to be like, what is that? Okay, elongation. So how are our cells getting longer? So like all science, we learn things through research. Research is how I am telling you what I know now and how you know what the Golgi apparatus is and, how we have electricity. And so let's tell a story through research. So we have some E. coli. And then we add an antibiotic called methicillin. So plus methicillin. So this is like a short time later. What happens to them? We noticed that this E. coli turned circular. So that has something to do with elongation. That's kind of strange. Uh-oh. And then they died. Did. Later. Okay, so what happened? What happened in this time? Why did they stop elongating before they died? Methicillin is an antibiotic, so they were going to die eventually. But what was happening here? So if these E. coli had not yet died from the methicillin and they were spherical, what if we found resistant bacteria? What if we found E. coli that were resistant to methicillin? So they did that. So these are wild type E. coli. These are our normal E. coli. But when we took mutant E. coli, we found that they were spherical. Interesting. And this isn't just on methicillin-based media. This is just like in general. So now we found these spherical E. coli and their mutants, so we can look. What are the differences in their genome? Why is it spherical, and what genes are responsible for this phenotype? What genes make it spherical? Huh? Okay, so how would you do this? Well, we learned before about complementation, right? So if we put in different wild type plasmids in our mutant E. coli, mutants, plus, I'm just going to say we have a wild type plasmid, a bunch of different types of wild type plasmids. So I'm going to say A, B, C. They probably tried more than 3, I will say. And eventually, maybe most of them didn't work. Maybe most of these different plasmids that we tried didn't work. But eventually we restored the wild type phenotype. So we could see, maybe that was from plasmid C. We could see what genes on this plasmid were responsible for the elongated phenotype. So eventually we found that one of these mutants, one of the many mutants we generated from the methicillin plating, right? We probably mutagenized the bacteria, plated them on methicillin media, and saw which one survived, and then we didn't just look at one colony. We found many, and one of them had a point mutation in a gene called MREB. And MR stands for methicillin resistant, and I don't know what E stands for. Maybe resistant. I don't know. And so that is interesting. So what does MREB do? MREB, which is italicized because it's a gene. Let's zoom out. Video skills go crazy. So MREB actually uses ATP to polymerize into something really similar to actin. So this is through polymerization. So if you're not familiar with actin, it's a component of eukaryotic cell cytoskeletons. It just makes that very rigid structure, but we don't have those in bacteria. But if we're looking at the cell wall, it makes sense to have something kind of like this actin material that can maybe strengthen our cell wall. So this congregates in patches. In the lateral cell wall. And obviously, researchers wanted to see what happens when you knock out this gene from bacteria. And we go from our nice normal E. coli, so MREB knockout. We have nice normal E. coli. They're all happy. And then they start to divide, but then they can't elongate. So they look kind of weird. And then they try to elongate, but instead they just look like lumps, and they're kind of gross looking, and some of them are I don't know, kind of freaky. Oh, I guess there should be more of them. We made 6. Where did the other 3 go? They died. Because they eventually do lyse, which I guess, next step. Lysis. This might not be exactly the scale, but you get the idea. Something is happening with elongation. So without our MREB, which can use ATP to become an actin-like filament, which is important for the lateral cell wall, we don't get that nice elongation. But hopefully you just caught a mistake in this experiment. If we knocked out MREB and this killed the cells, how did we grow these cells to begin with? Well, this is actually used with an inducible promoter. So first, we had it, and we had the knock. We made the mutants. We used an inducible promoter to grow them with, like, it induced. And then when we wanted to start the experiment and see what happens, we stopped inducing it. So that's how we had enough cells to observe for this. Because if you have one cell, maybe you made your mutant, but you can't see like what happens with that one cell. And even if you do, that's not a very good sample size. So we have a whole sample, a whole massive colony of millions and billions of bacteria. Okay, I don't know how many bacteria, but lots of bacteria, plenty to look at under a microscope and say, wow, it eventually killed them. It wasn't an immediate loss of function, but eventually they got all bloody. And in addition to testing these knockouts, they also tested a drug that T-polymerized MREB called A22. And that mislocalized our lateral wall synthesis proteins, like PPP2 and other ones, which we fortunately don't have to memorize. Lateral wall synthesis proteins. So, very important, it seems. So it looks like in addition to making these actin-like filaments, it has a greater role in directing PBP2 and other lateral wall synthesis things to making that lateral wall for elongation. And this doesn't deplete MREB. But we can see that when it's not polymerized in these actin-like filaments, the PBP2 and other stuff don't know where to go, or they're in the wrong place. And so even when it's there, if it's not polymerized, it doesn't work properly. So normally in these wild type cells, this one is looking very beany. The PBP distribution would be like mostly in the middle. So is this color going to work? That's fine. Would be like all throughout the cell, kind of evenly dispersed. So you can't really see the difference in these colors, but you will in a sec. So we'll say this is our PBP2 distribution. And so when you add A22, well, you get those blubby cells again. So it's more round. I guess I can make this one longer. You can see the PVP a little differently. But this time, it all concentrates in the middle. So something is wrong. They don't know what they're doing. PBB2 distribution is all focused in the middle. That's kind of weird. So turns out this one also makes a complex like a longosome. Sorry, I can't multitask. A longosome. It makes this complex called the elongosome. Like FCSZ and our divisome, we have the elongosome, which it's pretty well named. And I mean, we can say MRAB's complex. I'm not going to tell you what it does. I think it's pretty clear. Okay. And this synthesizes peptidoglycan in lateral cell walls. Walls. Okay. And so in addition to MREB and PBP2, we also have Rod A, Murray, and Merg, because you still got to be making your NAM and NAG subunits to make your peptidoglycan. Okay, so how do I get out? Let me out. Awesome. Elongation, not too complex. The history behind it is by looking at methicillin-resistant bacteria and seeing how they were spherical. See, and we were like, why is it spherical in our mutants? So okay, the mutants are separate from this is like our first experiment. Let's distinguish that. This is just with the wild E. coli, and after a while they died, but for a while there they were spherical, and so somehow they found resistant E. coli and found that they were spherical, and then they looked at them and found a gene that was responsible, MREB, which makes an actin-like filament, and that's important for directing PBP2 and other lateral wall proteins for being in the right place. So, and that was figured out by being depolymerized by A22. I actually want to highlight it in that color. Oh, and we also saw by using an inducible promoter, like, yes, that was important. The MREB knockouts did not survive. It was something to do with the lateral cell wall generation. So yeah, interesting. We have the elongosome made up of these lovely enzymes, PBP2, Rod A, Murray, and Merg, which we've all talked about before, and obviously makes peptidoglycan in lateral walls. Okay, we're going to use another page. I know, third page, one lecture, don't worry. It won't be long. We're not even that long into this video. And like FTSZ, it is important, like it needs MREB to form the elongosome. That's an important note. Okay, and that's why they're all dislocalized, is because they can't be part of the elongosome in a normal way. Okay, on to our last part, curvature. I'm going to write it curvy, curvature. Okay. And unlike the other ones, this one's actually not essential for survival. The way we found it was in transposon mutants. The way we found it was in transposon mutants, and they were alive and kicking, but they were normally curved. So this was actually in Caulobacter, which if you're taking microbiome right now at UC Berkeley, that's your professor. So Caulobacter. This is our wild type. And then transposon mutagenesis. I don't know what they were looking for, but what they found was some weird looking Caulobacter. Okay. It looked like this. They were straight. What the frick? How'd they do that? Why'd they do that? And why are they doing just fine? Like, is this okay? Are we okay? So they had to figure out, like, why is that? What happened there? What is responsible for that? And they found a filament called crescentin, and it's located on this inner edge. Crescentin. And this actually acts here, Cress. This is how we'll talk about it. This actually acts with MREB. So I'll just put this in a thought bubble in the corner here. It needs MREB to induce curvature. I'm going to say to make curvy. So people think, does it change the activity of the elongosome? Does it use the elongosome? I don't know, but that's the thought. Possibly. TBD, I guess. I don't know. Read some research papers. So I don't have to. But Crescentin, this is cool. We found a thing. And also, it's maybe I should like expand this line just a little bit because it's actually on the inside of the cytoplasmic membrane. I'm just gonna say inside of bilayer. So it's not on the outside, like with the cell wall, it's on the inside. But there actually is another one that's on the outside. In cholera, you could say Vibrio cholerae, a similar protein called CRVA acts in the periplasm. So this one is in the cell wall. Is closer to the cell wall. So that's interesting. So crescentin is in the bilayer, but CRVA is in the periplasm. Okay, so CRVA forms a complex with another molecule called CRVB. Crazy. And together they can actually make straight cells curved. So if you have a straight cell, it could be wild type straight cell, or you can make a curved cell straight, like you can mutate a different gene like CRES. And if you add CRVA and CRVB, it'll turn curvy. Which is crazy. Okay, actually, I just checked. They did this with Colobacter crescentus. So they did this with the normal crescentum one, the one that normally uses crescentin instead of CRVA, and they made it curvy, and they also used it with Agrobacterium tumefaciens. If you're in plant biology, you might know her, and I guess she's not curvy, and then they made her curvy, which is crazy. I should probably say with CRVB. So how does this work? How do they make it curvy? So researchers did the same thing they did before, where they like use two different colors to label the cell wall, and they saw that the inner curve divided less than the outer curve, which causes this like rounded bean-shaped phenotype. So outer curve grew faster than inner curve. And I don't know how this happens, but CRVAB binds in this inner curve. And if you were to take it away, so minus CRVAB, you get your outer curve and your inner curve are the same length, so they turn straight. Oh, this is not a very connected bacterium. So wait, what's the point? If we can survive without curvature, then why do we have it? Well, the idea is in really thick media, curvature actually helps them swim when it's thick. So they tested this initially in agar, but then they also tested it in like an intestine fluid, and which makes sense because it's where bacteria probably will be. And they saw that it helps them swim. And yeah, that's basically it. I told you this is not too long. Curvature, only half a page. Sorry, I hate to disappoint. I know you were really excited to learn so much about curvature, but it's not essential, really. Like you just really got to swim through thick media if you want curvature. That's the only reason you really need it. And how do we find out about curvature? We found out about it by using transposon mutagenesis. Remember that? Remember her? We found some crescentin, which is in the inside of the bilayer, unlike CRVA, which is in the periplasm, and it makes a complex with CRVB, turns straight cells curvy, turns without it, it makes curvy cells straight. Outer curve grew faster than inner curve, and it uses it interacts with MREB, and we think it has something to do with the elongosome. Cool. Okay, curvature. Do you remember everything we learned about before? Do you remember division? Do you remember her? We have our different shapes. We use some temperature sensitive mutants to see how cells made the septum, and we have all of these lovely things. Okay, Flash, do you remember them? Do you remember them? I'm gonna check. If I check, you see it too. I'm repeating. We're getting it ingrained in our brain here. We have the divisome, which is formed by FTSZ, and we have our transglycosylase, FTSW. Do we remember that from before? Do we remember our transpeptidase, PBP3, which is encoded within FTS1? Do we remember FTSA and ZipA, which attach the complex to the membrane? Do we hopefully remember these? I feel like this is a classic. You got to remember your steps of PG synthesis. And FTSK, which helps the dividing cell divide its DNA evenly so we don't have more DNA in one cell than the other. Which I feel like more DNA is not nearly as much of a problem as less DNA. I definitely don't want to be missing a chromosome. Anyway, I want exactly as many chromosomes as I have though. We can also get longer, very important. We know MRIB is really important in that, and it makes the elongasome. And with A22, we were able to see what happens when we have MRIB, but it doesn't polymerize, which is our lateral cell wall things get all confused. They're in the middle for some reason. Why are they there? We're supposed to be growing everywhere, not just dividing over and over again into whatever that is. And then they die, which is sad. Very sad. For our colobacter, they're not even pathogenic. Okay. Goodbye, guys.